So proteins, that's the molecule formed by RNA translation. Um, so, like I said, proteins are polypeptides formed by amino acids through peptide bonds by the ribosome. The template is initially DNA and then finally mRNA before it's turned into a protein by the ribosome. And proteins have a wide variety of functions in our body. So some of the functions are, it can be an enzyme that catalyze biochemical reactions. So they form enzymes that speed up reactions without being used up. They decrease the amount of energy needed for chemical reactions in order to speed up the reaction. Or they're peptide hormones that are used for chemical signaling over long distances. And examples of those are insulin and glucagon. So these are peptide hormones. I know most hormones are steroids, but there are a few peptide hormones that are used. And then proteins, a main function of them are structural proteins. So they give shape to the cells and they're used for movement. So proteins like actin and myosin, they're used for movement. They're in our muscle. They're used by the cell for movement. And so, like I said before, the proteins, it needs a specific shape in order to function. So when denaturation happens, it disrupts the protein shape and the protein loses its function because of that. So proteins, they need a specific shape in order to function in our body. And it can happen because of changes in temperature, pH, or specific chemicals called denaturants. So if a protein gets too hot or too cold, it'll lose its shape and lose function. If the pH is with it outside of its optimal range, it'll also undergo denaturation. But denaturation, it causes the proteins to lose its 3D shape. So it won't change the amino acid sequence. So the primary structure, which I'll go over in a bit, it doesn't lose the amino acid sequence created by the ribosomes. It just loses the shape it adopted after RNA translation. So all the post-translational modifications that it had in order to get to the proper shape, it loses that. But if the change that caused the denaturation is removed, it can regain its 3D shape again over time. So the primary structure, like you said, it's the actual identity of the protein. It's determined by the chain of amino acids linked together by the ribosome and predetermined by the mRNA that was transcribed. So like I said before, they're stabilized by the covalent peptide bonds between the adjacent amino acids. So the primary structure gives the actual identity of the protein. So different primary structures lead to different proteins. And then the secondary structure. So the secondary structure is formed by adopting the most energetically favorable arrangement of the primary structure. So the primary structure is linked by peptide bonds, but the secondary structure is mostly determined by hydrogen bonds. So hydrogen bonds between the different amino acids, they provide stability. They form these two structures on the right. So you see primary is just the chain of amino acids, but then secondary, they form these alpha helices on the left, and then on the right are these beta pleated sheets. So these are formed because of the stability provided by the hydrogen bonds. So the carbonyl oxygens and the amide hydrogens of the different amino acid residues, they hydrogen bond in order to form these specific secondary structures. So the primary structure is just a chain, but that's not very energetically favorable. So they adopt these two secondary structures in order to form hydrogen bonds between them, and that provides more stability. And then the secondary structure can be disrupted by denaturation, but it can reform these secondary structures if the denaturant is removed. And then tertiary structure. So this tertiary structure also adds to the 3D shape. It is formed by hydrophilic and hydrophobic interactions between the R groups. So each amino acid has a different R group. That's like the side chain that's attached to it. The side chains, it may be hydrophilic, which in that case, it will be on the outside, and it will form hydrophilic interactions, while hydrophobic groups, they don't like water, so they'll be more likely be on the inside of the tertiary structure. So it's more likely to find hydrophobic R groups in the center of the protein, while the hydrophilic groups will be on the outside. And these are all determined by the R groups connected to the amino acids. So hydropolar and charged amino acids, they're more likely to be on the outside, while hydrophobic R groups will most likely be on the inside of the protein. And then another thing that adds to the 3D shape are disulfide bonds or disulfide bridges. So this is when two cysteines are oxidized to form cysteine. So two cysteines combine together in order to form a disulfide bridge, and that also adds to the 3D shape of the protein. And disulfide bridges, they can also be broken apart by denaturation, but then also reformed as well after denaturation. Denaturant is removed. And then finally, the coronary structure. So not many proteins have coronary structure. This is only when the protein has more than one polypeptide chain. So this is when the protein has multiple polypeptide chains combined together into a coronary structure. So this is formed by aggregating the different protein subunits into a coronary structure. So like hemoglobin would be an example where there's multiple subunits that combine together in order to form the full protein. And